Preclinical development represents a crucial phase in the development of pharmaceuticals, where extensive testing and evaluation are conducted before a potential drug candidate can progress to clinical trials in humans. One of the essential components of preclinical development is the assessment of toxicity. Toxicity studies aim to determine the safety profile of a drug candidate and its potential adverse effects. In this short video, we will delve into the main toxicity studies undertaken during preclinical development, highlighting their significance and impact on drug development. Acute toxicity studies assess the immediate adverse effects of a drug candidate when administered at high doses to animal subjects. Acute toxicity studies provide valuable information about the potential risks associated with a single, high-dose exposure. Researchers use these studies to establish the drug's maximum tolerated dose, MTD, which informs dosing strategies in subsequent studies. Unlike acute studies, subacute and subchronic studies involve the administration of the drug candidate over a longer duration, typically ranging from a few weeks to several months. These studies help identify adverse effects that may manifest over time, providing insights into the drug's safety profile during repeated dosing. Subchronic toxicity studies are particularly important for chronic conditions where long-term drug use is expected. Chronic toxicity studies extend the evaluation period even further, often spanning a significant portion of an animal's lifespan. These studies are critical for assessing the potential risks associated with prolonged drug exposure, which is essential for drugs intended for long-term treatment, such as those for chronic diseases like diabetes or hypertension. Reproductive and developmental toxicity studies evaluate a drug candidate's impact on reproductive health and fetal development. These studies assess fertility, pregnancy outcomes, and any adverse effects on the developing fetus. Ensuring the safety of medications for pregnant women is of utmost importance. Genotoxicity studies examine whether a drug candidate has the potential to cause genetic damage or mutations. These studies use various tests to assess the drug's impact on DNA, chromosomes, and genetic material. Identifying genotoxicity early in drug development is crucial, as genetic damage can lead to serious health concerns, including cancer. Carcinogenicity studies investigate whether a drug candidate has the potential to induce cancer. These long-term studies are often carried out in rodents over their lifetime to assess the drug's tumorigenic properties. Identifying carcinogenicity risks is vital for ensuring patient safety, as cancer is a severe and life-threatening condition. Depending on the nature of the drug and its intended use, additional specialized toxicity studies may be necessary. For example, specific studies may be conducted to assess organ-specific toxicity, immunotoxicity, neurotoxicity, or cardiovascular toxicity. These studies tailor the evaluation to the potential risks associated with a particular drug. Safety pharmacology studies focus on understanding the effects of a drug candidate on vital physiological functions, such as cardiovascular, respiratory, and central nervous system function. These studies help assess whether a drug candidate may have adverse effects on these critical systems. The results of these toxicity studies are pivotal in determining the safety margin and risk-benefit profile of a drug candidate. If significant toxicity is identified during preclinical development, it may lead to the termination of the development program or the need for further refinement of the compound. In a nutshell, toxicity studies conducted during preclinical development are instrumental in ensuring the safety of pharmaceuticals before they enter human clinical trials. These studies cover a range of durations and endpoints, from acute toxicity to specialized assessments, providing a comprehensive understanding of a drug candidate's potential risks. By identifying and addressing toxicity concerns at the preclinical stage, pharmaceutical companies can enhance the likelihood of developing safe and effective treatments for a variety of medical conditions, ultimately benefiting patients and healthcare systems worldwide.